Hello, my name is Lynn Gilbert. I'm an infectious disease physician and clinical microbiologist. This talk is a collaborative effort between myself and my colleague Vitaly Sinchenko, who is a clinical microbiologist and public health epidemiologist. In it, I will describe some methods of pathogen strain typing, and in particular, explain the uses and benefits of pathogen whole genome sequencing for prevention and control of infectious diseases. First, I want to start by defining what we mean by a species. This is a group of organisms that share genetic characteristics to the extent that they can exchange genes, such as by reproduction. Members of the same species, humans for example, Homo sapiens, or dogs, Canis familiaris, can look and behave very differently despite close genetic similarities, or they may appear indistinguishable despite significant genetic differences. Similarly, microbial pathogens of the same species can rapidly develop genetic differences that are not obvious based on how they look or behave, but they distinguish them as different strains. And this is important because when an outbreak of infection occurs, it's usually due to a single strain of a microorganism. And in order to investigate the source and spread of an outbreak, we need to be able to identify all of the cases that are caused by the same strain and exclude any cases that are caused by different strains. We do this in the laboratory by using various strain typing methods, which these days are usually based on genomics. To illustrate this, imagine this is a hypothetical graph showing the numbers in each month of patients in a hospital from whom the common hospital superbug, methicillin-resistant Staph aureus, or MRSA, was isolated. In order to reduce the spread of MRSA from one patient to another, we need to be able to understand whether they belong to the same strain or different strains and how they spread from one to another. Strain typing of isolates from this hypothetical study in fact shows that there are several different strains shown in different colours. If we separate the main strains out into two separate graphs, we see that there are separate outbreaks, each of which is likely to have a different source and in much more manageable numbers for investigation. There are many different strain typing methods. For example, we may select a panel of genes that we know are present in some but not all members of the species, and use probes to show which ones are present or absent in different strains by the pattern of the probes that react with each strain. If the patterns are different, as shown in this diagram, mostly, we can infer that they're not related, although there are two in this group that are indistinguishable. But if some or all of the isolates are indistinguishable from each other, this would lead us to suspect that they're part of an outbreak. An alternative method of strain typing is to choose a single gene which is present in all strains of the species that we're investigating, but not in other species. And these genes can vary from one strain to another without changing the function of the gene. By sequencing that gene from each of these strains, that is, we show the order of the DNA building blocks or nucleotides, which are designated as A, T, C and G, we can compare the sequence of nucleotides in different strains. And again, if a sequence from different isolates show significant differences, we can infer that they're different strains. But if they're identical or extremely similar, they may be part of an outbreak. But still, we can't be absolutely sure. Because these strain typing methods that I've already mentioned are limited by the fact that they only analyse very small proportions of the whole genome. This means that different strains may appear indistinguishable or closely related, but actually have significant differences in the parts of the genome that have not been analysed. So we may get a false impression that they're part of an outbreak. And in fact, investigating a false or poorly defined outbreak can waste a lot of time and still not identify a common source typing methods. This can be illustrated by imagining that we have 5 people, each of whom has been infected with the same pathogen species. Let's use MRSA as the example again. If strain typing using the panel of genes that I mentioned before shows that isolates are different, we can be confident these people are not part of an outbreak. But if their isolates are indistinguishable, we'll suspect an outbreak, but we can't be sure because, as I said, there may be significant differences in parts of the genes, parts of the genome other than those selected genes. And of course, even if they are all part of the same outbreak, this strain typing method can't tell us how or in what order the outbreak strain spread from one person to another. This is where whole genome sequencing comes in. If we sequence the whole genome, we can identify small differences in individual nucleotides between isolates and construct a family tree that shows how closely related they are. Isolates from the same outbreak, which have either identical sequences or very few, usually less than 10, we accept as being close enough to being the same strain, cluster together in the family tree, and they cluster separately from unrelated isolates. And as well as showing how closely isolates are related and whether they belong to the same strains or not, whole genome sequencing can identify certain genes in the outbreak strain that may affect the outcome or be important in treatment of patients, such as antibiotic resistance genes. Let's go back now to our hypothetical outbreak, and we can illustrate this better by looking at a very short segment of the whole genome sequence where this small number of nucleotide differences occur between these different strains. Isolate W in the top row has none of these mutations, so it's the first in the series. Isolates E and K have each arisen independently from isolate Y by acquiring 2 and 1 mutation respectively. Isolate E hasn't spread to any of the others in this group, but isolate K, as you can see, by picking up another mutation, has spread to become isolate W, which in turn has picked up a third mutation to become isolate G. By showing these relatively minor differences, we can map the spread of MRSA from one person to another. Now I want to illustrate how pathogen whole genome sequencing can and has been used to investigate outbreaks of infection. The first example I want to use is of an outbreak of MRSA infection in a neonatal intensive care unit. Over a 2-day period, 2 very premature infants who were nursed in the same 2-cot room developed severe sepsis and died from it. Routine strain typing showed very quickly that both were infected with the same rare strain of MRSA. So it was very urgent then to determine, if possible, where this strain had come from and whether any other babies were at risk. So we screened all staff and patients in the unit, but unfortunately were not able to identify any who were colonised with MRSA. Nevertheless, so that we could identify any future ongoing risk as quickly as possible, we introduced routine screening of all the babies at the time of admission, usually immediately after birth, and every week while they remained in the ward, often for many weeks, as you might imagine, with very premature babies. And of course, at the same time, routine infection control measures such as hand hygiene and environmental disinfection were tightened to reduce the risk of transmission. Despite all these measures, the new strain kept on showing up in new babies. Over the course of the 10-month period, 14 new babies were colonised, and some of them developed significant infections. Each horizontal bar in this diagram represents one of these babies from the time of admission to discharge. The yellow portions of the bars indicate the period after they were first screened for MRSA but found not to be colonised, and then the red portions of the bars show that they were screened when they were screened again and became colonised. And as you can see, many of them were in the ward for quite long periods of time, while they remained potential sources of spread to other babies, possibly via the vector of the contaminated hands of staff or parents. Because of this risk, staff were screened again some months after the initial screening, and this time one nurse, who we'll call Nurse N, was found to be colonised. Soon after that, another nurse, who was also a staff member in the neonatal intensive care unit, who'd been negative during the second round of screening, presented to the emergency department with an infected abrasion on her leg, from which the same virulent MRSA outbreak strain was isolated. After her infection was treated successfully, the outbreak stopped. No more babies were colonised or infected. And these results raised the possibility that one or other of these two nurses, and particularly possibly Nurse NE, might have been the original source, had been missed by one of the two bouts of screening, or else a vector of the MRSA strain. This question was potentially answerable by whole genome sequencing, but to do that, and this was only considered some months after the outbreak had ceased, raised a number of ethical questions. The aim, of course, was not to blame anybody, but to try to understand what had happened so that it could be prevented in future. However, we couldn't exclude the possibility that if someone were identified and knew that they had been an unwitting source or vector of spread, they may feel very guilty. And if that information was leaked to other people, they may be blamed. So the question was whether we should seek consent from any of the people from whom these strains had been isolated. Because it occurred sometime later, we chose not to do that and not to share the results with either the nurses involved or the parents of the affected babies. In fact, whole genome sequencing showed some useful results. First of all, Nurse N, shown up here at the top, was neither the source of the outbreak nor had she spread it to anybody else. In fact, she had acquired MRSA from 3 babies, who were triplets in fact, whom she had been nursing for quite a long time. Secondly, it showed that Nurse NE was not the source. Her isolate had arisen from the original baby, but she acquired the MRSA strain quite early in the outbreak, at the same time as a number of other babies had. And it's possible that she spread this strain unknowingly during the 10 months while new babies were becoming colonised, particularly since the outbreak stopped abruptly after her infection was treated. However, it's also quite likely that at least some instances of transmission were due to the contaminated hands of other staff members who had not been identified by screening either. Another important application of whole genome sequencing is in salmonella surveillance and outbreak detection and investigation. Salmonella is the commonest notified cause of foodborne illness in Australia, with an estimated 40,000 cases and 2,000 hospital admissions each year, and a not insignificant number of deaths from infection, particularly in very young infants and the elderly. And despite intense surveillance over the past many years and improved strain typing methods in recent years, the incidence has continued to increase. What happens when an outbreak of salmonellosis is suspected is that public health officers will follow up selected cases who they suspect to be involved in outbreaks. They'll take food histories and ask about where people have eaten, where they obtained their food from. And if some types of food or food outlets, such as shops, restaurants, or a particular event like a wedding, are shared between different members of this people involved in this outbreak, environmental health officers will inspect the premises, take samples of food to see whether there's evidence of salmonella contamination. The problem is that once an outbreak is confirmed, it's often too late for people to remember what or where they ate, and any residual food that's left at the premises is likely to have been discarded, so it's too late to intervene to limit the number of cases or prevent another outbreak from the same source. The reason it takes so long to identify or confirm an outbreak is that until recently laboratory investigations have been very slow. Salmonella come in at least 2,500 different serovars. They're highly variable organisms, and each of these serovars can differentiate into a number of different strains or subtypes. Strain typing is a complex business, and it can only be done in reference laboratories, which means that the strains have to be sent to a laboratory other than the one that originally diagnosed the infection. And at least 2 different strain typing methods are required to identify particular strains, and this takes up to 3 or sometimes even more weeks before the final result is known, which is often too slow for an accurate food history to be obtained. However, since 2016, the reference laboratory in Sydney and some others in Australia have been performing whole genome sequencing routinely on all salmonella isolates, and the results are available within a week or even a few days of diagnosis. This provides all of the information that the previous strain typing methods provided, but it's much faster and less expensive. And it also means, of course, that a suspected outbreak can be confirmed, and often otherwise unrecognised outbreaks can be identified quickly enough that food histories are more accurate, a suspected source of the salmonella can be identified and confirmed in time to prevent additional cases occurring. It also means that sequence data can be shared between reference laboratories in different countries, or, for example, in different states of Australia, to identify national or international outbreaks due to food products, which, as they often are, have been widely distributed around the country or from one country to another. Now that food producers, distributors and retailers know that if they have poor food handling practices or production practices, they're likely to be identified if they cause a foodborne outbreak, it puts them on notice, hopefully to provide a very strong incentive for them to maintain the highest possible standards of food handling practice so as to avoid litigation and significant threats to their business. And of course, if so, this will lead to fewer foodborne disease outbreaks. Whole genome sequence data can also track specific virulent or antibiotic resistant microbes, and in particular this is an example of another salmonella strain between countries by sharing data in international databases. This diagram shows the family tree of isolates of a particular multi-drug resistant salmonella strain in 4 countries, including Australia, which is where the isolates are shown in yellow. By demonstrating how these isolates cluster together over time and in different countries, it was possible to show in this investigation that the isolate was probably, the strain was probably introduced into Australia from the United Kingdom, which are shown to the right of the Australian strains in green, but that it was probably spread locally, often from person to person rather than necessarily by food, rather than multiple separate introductions. And because some of these isolates included isolates from food products, it was also possible to show that pork meat was probably the likely vehicle by which it had been introduced. International International surveillance such as this uses de-identified data. Only the specimen and date of isolation are shared internationally. But local outbreak investigations, although provides crude information, just transmission over time and different geographic locations, but outbreak investigations necessarily means that public health officers, in order to obtain food histories and understand how spread occurs, inevitably must have identifiable information, know who the people are who are involved in outbreaks. This diagram illustrates the trade-off between the usefulness of pathogen whole genome sequencing and related demographic data and privacy. As we've seen, pathogen whole genome sequencing provides far more personal information than previous methods of strain typing: where and when the person became infected, who or where the pathogen came from, and who it was passed on to. This may be information that the person from whom the pathogen was isolated is unaware, or may be regarded as private, especially, for example, if it happened to be a sexually transmitted pathogen, for example. Every additional fact, such as gender, postcode of identification, postcode of address or age, that's added to the information, such as whole genome sequencing, provides much more useful information from the point of view of outbreak investigation, but it also significantly increases the risk that the data can be de-identified, and therefore a potential risk to privacy. So, in summary, pathogen whole genome sequencing has many potential benefits. It can identify the scope of an outbreak, it complements epidemiological investigation, and it can identify characteristics of an outbreak strain and a single reaction that may be important in management of the outbreak or of individual cases of infection. It can show the sequence of transmission and identify the source. But its effectiveness depends strongly on speed. We need, for this to be useful, we need rapid routine whole genome sequencing to be performed and analysed as quickly as possible so that public health action can be implemented very promptly. However, if this occurs, routine whole genome sequencing of pathogens of public health importance will mean fewer illnesses and deaths from infectious diseases. Nevertheless, these benefits come with significant risks and ethical issues. Because of the need for speed and routine sequencing, individual consent from people from whom the isolates came is impractical. And that's why we have public health legislation which enables this to occur with all of the appropriate safeguards of privacy. But it doesn't cover all instances of whole genome sequencing. This information has potential commercial or legal implications. If a food outlet or a hospital, for example, is identified as being a source of infection, there are risks to business or the livelihoods of people who work in them. And there are medico-legal or occupational safety implications. If a staff member is found to be a source or vector of infection, is the staff member at fault, or is it the hospital system at fault? And is there any fault at all? It's a problem of many hands that we can't necessarily identify exactly what goes on. And if this information is misused in any way, if it's accidentally leaked or hacked, there are personal privacy issues, stigmatisation of the individuals or perhaps groups of individuals who are involved, and even to their career prospects. So to summarise, finally, better data with data linkage will certainly improve communicable disease control, but it reduces individual control of information and risks to privacy. So we need to move with great caution and sensitivity in using and generating these data. Thank you.